It's a pleasure to, well, be here, but talk to you about the pancancer compendium of chromosomal instability. But before we get started, and because I can't be where you are, can't even move my slides. This is my Cambridge. It's Cambridge, UK, not Cambridge, MA. We consider your kind of Cambridge the kind of young upstart. Well, not too impressed. We're much older. What you see here is the River Cam, and there's a bridge in the back. So that's pretty much Cambridge. This is the picture card view of Cambridge, and it's not where I work. I'm here. This is in the south of Cambridge. This is the biomedical campus. Old Cambridge, where this other picture was taken, is somewhere in the back there. It's very far away. When traffic is slow, 10 minutes by bike. We'd never go there. But we have all this kind of research stuff here in the south. There's an institute called the LMB. This is the one Watson and Crick have started. They, back in, I don't know, 50 something, have 20 faculty, except for 2. Everyone has a Nobel Prize. So if you ever suffer from imposter syndrome, think of these 2 group leaders surrounded by 18 Nobel Prize winners. It's not good. The thing in the middle is the new AstraZeneca headquarters. So we have a big pharma company sitting here, but the rest of what you see is all hospitals. So there's Pepworth, which is a lung and respiratory hospital, and then Addenbrooke's, which is one of the biggest and advanced hospitals in the UK. And now right in the middle of all of this, there is the Cancer Research UK Cambridge Institute. And if you look very closely to this one window on the 3rd floor over here, you can see me waving. Just look closely. You can see me waving out of that window. That's where I sit. So the reason I show you that is I'm a mathematician by training, but you see, I'm just right in the middle of a biomedical campus, very, very close to the hospital. And while computational stuff is at the core of what we do, all the goals, my aims, the reason I do this is now cancer research and trying to find biomarkers, find drugs, find stuff to help people. My group does pretty much everything. The only thing we do not do is focus. There's 3 different parts to it. What I will tell you about today is mostly from Team Genomics. Actually, it's only Team Genomics, but we also have an imaging team where people look at radiology and pathology images and ideally combine them with some genomic information. And I have an experimental team, that's Team Networks, where we look at perturbations of cells and from the effects try to figure out how some key mechanisms like the estrogen receptor actually work. But for today, what I will be talking to you about is all genomics based. Last thing before we get started, I have a conflict of interest to report. Based on the work that I'm presenting you today, we started a company a couple of years ago. So you guys need to be extra careful with everything I say, because that could just be a sales pitch. All right, so the key word, the key concept to introduce, what's in the title already, is chromosomal instability. And chromosomal instability in a way just means completely chaotic genomes, genomes which are a complete mess. So what you see here in this little movie is about 120 high-grade serous ovarian cancers. And ovarian cancer is one of the best known highly chromosomal unstable cancers. You see on the x-axis, you see the chromosomes. And on the y-axis, you see the absolute copy number, like the number of pieces of DNA of a particular locus. And if you can spot a system or a pattern in there, that would be amazing, because we can't. There's very few recurring changes. There is almost no patterns that we can use to predict anything for these patients. This is just very, very chaotic. Technical note, while we look at this, what we're actually looking at is aneuploidies. All the data you see comes from bulk sequenced patient samples. So we see kind of stable DNA changes, whereas if you become very specific, chromosomal instability is actually the process by which you arrive at these stable changes, and we'll come back to that at the end of the talk. Now, if you look at this chaos, this is what we try to understand, and we try to dissect it and make sense of it. There is a big medical need. Many of the very lethal cancers, ovarian cancer, pancreatic cancer, brain cancers, lung cancer, have not seen any advances in treatment or diagnostics for many, many decades. And that is because they're highly chromosomally unstable, very few recurrent events. You just can't make sense of these genomes. Now, lots of people have studied chromosomal instability, and they focused on two different areas. Many people looked at the consequences of chromosomal instability, and there's lots of things to discover. Chromosomal instability leads to loss or amplification of driver events, to focal rearrangements. Metastases are often much more chromosomal unstable than the non-metastatic cancers. Sometimes the DNA is so messed up, it leaves the nucleus, becomes extra chromosomal DNA, and then it even has an immune response in the cytosol. So there's lots of stuff people discuss as the consequences of sin. Other people, it's mostly other people, it's never the same people, look at the causes of sin. And so how do you get these completely messed up genomes? And some of them focus on mitotic errors and defects. So when you have cell division and the chromosome pairs get pulled into the two daughter cells, if something goes wrong and you can't get pulled over, you suddenly have an amplification, for example. Other people look at telomere crisis, when like the ends of chromosome get cut and then they stick together in ways they should not, which can lead to breakage fusion bridge cycles. And one of the most common form of sin that people study is called HRD, homologous recombination deficiency. That is, HR is one of the key repair pathways. If you have a double strand break, it uses a sequence homology to repair this. And if you're defective in this, then your DNA is really messed up. So this was very short, but the picture I want to paint is that sin is very often used as an umbrella term. It's just like this broad term to say genomes are messed up. But when you look into that, there's lots of nuance. There's lots of different consequences and lots of different causes. But what has been missing is good measures of sin. Measures that you can apply to a genome to figure out what caused it, what are the consequences, can we somehow relate these different types of sin to different clinical phenotypes and kind of break open this umbrella term and get more nuance to it. And this is what I'm here to tell you. That's the goal for today. The key tool we will be using are mutational signatures, which are indicative of different mutational processes. The key idea is that if you see a messed up genome and lots of genomic changes, then there is many, many different reasons or processes which could have led to this. I mean, in this picture, in this cartoon, I painted 5 of them. One of them is sunlight. So if you get out too much in the sun and you get sunburned, that's bad for your DNA. So wear sunscreen. Smoking is no good. Smoking leads to mutations, and you see that in your DNA, so don't smoke. Some things are harder to avoid. Aging leaves mutational marks in the genome. Then there might be stuff like HRD again, homologous recombination deficiency, which you might be born with because you have a mutation in the BRCA gene. And when we see genomes, you have to measure them. There might be just technical artifacts. Now, for any given genome, these different sources of mutations will not act at the same strength. So it could be that in one patient, they are HR positive, but they are strong smokers. And that will leave particular marks in the genome, which might be very different from the next patient who has never smoked, but is HRD. So the idea of mutational signatures that I want to introduce is to act as the characteristic fingerprints of different mutational processes. And if I now look at a genome and I know how these fingerprints look like, I can say, well, that's the genome of somebody who has no HRD but was a smoker. And the other genome, that's a genome of somebody who has never smoked but has HRD. Problem, of course, is a priori, we never know what these characteristic fingerprints are. Somehow conceptually, they are genomic patterns. They somehow correlate or are indicative of a mutational process, so they have a biological meaning, but we don't know how they look like, so we have to discover them. And there's a whole field out there that does mutational signature analysis. Serena Nixinal here in Cambridge, one of my colleagues, she's the leader in this field when it comes to single nucleotide variant mutations. And the people at the Sanger is a little institute in a little village, even further out here in the countryside, outside of Cambridge. They do lots of SNV signatures. And I'll give you the cartoon version of how signature analysis works, and then I will apply it to our problem of understanding chromosomal instability. So mutational signature analysis in the cartoon version looks like this. You are given hopefully large number of genomes, and they all have some kind of mutations. Very often it's single nucleotide variants. In our case, it will be copy number changes. You stare at them very hard with your computational head on, and you somehow find things you can count in these genomes to map all these mutations into some kind of d-dimensional feature space. So the SNV people just count how often do I see A to G or G to Ts, and that's the kind of profile they look at. I will show you how we do this for copy number on the next few slides. But once you have taken your genomes and transferred them into a count space, you have a matrix of your N genomes in one direction and the features you have counted in the other. If you want signatures now, you just use some kind of matrix decomposition. The most popular method is non-negative matrix factorization, which will give you two matrices out of one. The green one, that is our fingerprints. It's a signature catalog. And the orange one tells us how active each of these particular signatures was to create the blue matrix. If you guys do gene expression analysis, this is the same kind of methods people use to cluster gene expression data. There is one particular parameter in there, which is, I marked it in red, it's the number of signatures. So this is something you have to choose based on model selection, which is a dark art. So most people just stick the finger in the air and come up with a number they really like. Now, so far, this is just mathematics. And whatever you count in these genomes, whatever your blue matrix looks like, if you do some kind of matrix decomposition on it, you will get two matrices at the end. So most of our job is actually signature interpretation. We try to figure out if the different signatures we found actually have a biological meaning. And there are two different strategies for that. And again, when I come to my own papers, I'll show you in detail how we do this. One of them is that you look at the green matrix, which somehow lives in the space of sequence features D, and somehow encodes how the genomes look like. And you compare that what you might know a priori or like from other experiments, how different mutagens change the genome. So if somebody, I don't know, smoked right next to a cell line and then afterwards sequenced the cell line, they might find certain changes in the genome, and they say, well, they must be smoke induced. So if one of my signatures, one of my S signatures has the same changes, you say, well, that might be a signature for smoking. This is in a way a direct way, which assumes that you have some experimental evidence specific for different mutational processes. The other way is a bit more indirect, and you look at the activities of the different signatures across the patients and correlate them with patient covariates. What do you know about them? Did they respond to a particular treatment? Were they smokers, for example? All kinds of correlations you can do. The first part of this I will call easy, because in a way it's just mathematics. I mean, it will just work. You will get these algorithms to run and you will get matrices out of it. The really hard part is the second one, where you make biological sense of what you just computed. And this is what we spent years on. And that's what I'm going to report now. So the first example of the signature analysis is a paper we published a few years ago. The first author is Jeff McIntyre, who is now a group leader at the Spanish National Cancer Center in Madrid. And there was a collaboration with Professor Brenton and Ian McNeish from Imperial and the second author is Dr. Goranova. So what we did in this project, we looked at ovarian cancers. And while other people had looked at single nucleotide variants, we wanted to understand chromosomal instability. So we tried to understand which mutational processes shape copy number. And the first step we had to do, remember the cartoon, we somehow had to come up with a way to encode copy number profiles numerically. And the way we did this is summarized in this slide. On the left, we start with absolute copy number data. So you guys know the difference between absolute and relative. So if you do copy number analysis and kind of what comes out of most of the analysis and pipelines is relative copy number. You often have a normal sample and a tumor sample, and the information you get is there's more DNA here in the tumor sample than there was in the normal. But you do not yet know how much that really is. Like, what is the absolute number of copies? And to get there, you have to correct for the purity of the sample and the overall ploidy. And it's a bit of a dark art, but it leads a lot of manual curation, lots of experience. So that's what you have to have done to get us started here. So we have absolute copy number profiles. They were, in our case, done from shallow whole genome sequencing. How shallow do you think can you do whole genome sequencing to still get copy number? I mean, this is 0.1x. This is very shallow. It's a very cheap assay, 50 pounds for us, which is something you can actually try to use in the clinic. You don't get poor from this. Now, from these profiles, and that's the middle panel now, we derived 6, as we claim, fundamental copy number features. Some of them are the density of breakpoints, either in 10 megabases or in whole chromosome arms. Some of them are the actual copy number, the height of some of these segments. A third one is the jumps in between consecutive segments, the copy number change points. One of them is the length of change of oscillating copy number. If you guys know what chromophripsis is, this is one of the kind of defining features. And some of it is just segment size. Are the segments long or short? And many of them we just came up with because if you describe a copy number pattern, this is the information you would use. I see lots of long changes at high ploidy. And some of them were handpicked, like the oscillating copy number, because we looked into what patterns people had described in genomes, and we tried to catch them, like, for example, chromophripsis. Now, this is not yet giving us something to count. These are just genome-wide distributions we have of these different copy number features. So we then applied mixture modeling to all of them, and we described each of the genome-wide distributions with a certain number of model components. And this differs for the different feature distributions we looked at. So what you see here, for example, is that the copy number feature is best, that's the second one, is best being described by 8 different components, whereas the segment size feature is the one at the bottom, is best being described by 10. If you add all of this up, you get to, I don't know, 36 or 37 different features. And that is actually what we use to count our genomes. So if you come in with a new genome and you say, Florian, can you embed this in our count space? I will look at the lengths, for example, of every segment. And I will say, well, I see component 9 represented very often in your genome, but component 1 never. And so component 9 gets a high count, component 1 of the segment size distribution gets a low count. And if you do this for all 36 components, you get the following matrix, which has our 117 tumors. That's actually exactly the 117 where I showed you this movie in the beginning in one dimension. And you have the 36 features in the other. And we just counted for each component how well represented it was in each of these tumors. That is the input matrix. And then we applied non-negative matrix factorization, which gave us 2 matrices. One is a tumor by signature matrix. It's the, in a way, the activity, or sometimes people call it the exposure matrix. It tells us how active each signature was in each tumor. And the second one, that's the fingerprint. This is the signature by component matrix. In our case, there's 7 signatures. and it tells you how each of these signatures is described by the 36 components that we have chosen. How did we come up with number 7? Very deep scientific insights. I mean, there are all kinds of quality metrics you can use for NMF. We did all of them, and in the end, we kind of convinced ourselves that 7 was a very good solution. This is really, if you want to repeat this, this is one of the very, I don't know, less quantitative parts, because you get all these metrics and you have to make sense of them. So for us, 7 was a good solution. Now I want to show you how one of them looks like. So let me go back. The first, if you look at the rightmost matrix, the first row in the rightmost matrix, that's what I'm showing you now in this bar plot. So the last one was a heat map. Now I show you the same information in a bar plot. What you see on the x-axis are the 36 different features. And the 36 different features come from 6 different distributions. And you see that signature 1 is, for example, not characterized at all by copy number, not characterized at all by change points, not characterized at all by oscillating copy number length, but is characterized by density of breakpoints and by segment size. That's the first hint of what's happening here. We then took the, well, this is good. This is not, didn't really tell us what to expect. But this is just gives us an overview of what kind of genomic pattern we are seeing here, and it's pretty much one with very long segments. We then correlated the activities of all 7 signatures, including the one you just saw, against all kinds of covariates we had for the patients. And that was, for example, you see this on the left here in this plot. It was from age of diagnosis, whether there were foldback inversions, how long telomeres were, how many chromophriptic events there were, all the way down to mutated pathways. Was the Wnt signaling pathway mutated, yes or no? And what you see is a picture. If you look at the first column of this matrix, you see the signature 1 again. And you see in purple that it was very highly correlated with foldback inversions. So this is a type of genomic event where 2 chromosomes, the ends, the telomeres, they're too short and where they fall off, and 2 ends of chromosomes stick together. And when they get pulled apart again, they're not getting pulled apart at the position where they stuck together, but somewhere else, and then the rest folds over. And that happens at the telomeric region, and it's kind of this long event, low breakpoint number that we saw here. If you go down the first column, you see it's in red. It's, for example, negatively correlated with the HRD signature people had described for single nucleotide variants. And that is another evidence that what we're looking for had something to do with foldback inversions, because these 2 events, HRD and foldback inversions, rarely co-occur. If you look at some of the other signatures, for example, signature 3, it's a very different picture. Signature 3, for example, is very highly correlated. So I'm looking into panel C, second row right now. Signature 3 is very highly correlated with the HRD SNV signature. And the patterns, they also look very different. So this is a putative HRD signature. So by putting all of this together, we had stories for almost all of our 7 signatures, what the underlying biology was. We also, what we could do is, where are my slides? Here. We looked at our 120 patients, and additionally, patients from PCOC and TCGA. Altogether, these are 575 patients. And we looked at, so in this colorful plot, each column is one patient, and the stacked colors is the activity of each of our 7 signatures in this patient. And the patients are ordered by the risk of death from a proportional, from a Cox proportional hazard model. So this was an analysis to see, we have some biology, but do these signatures actually mean something for the patients, like clinically? And so if they correlate with survival, that would be very interesting already. What you can already see by eye is that signature 1, the foldback inversion signature I showed you, there's lots of it on the left, but there's very little on the right of the plot. Whereas something like signatures 3 and 7, that's the purple and the brownish one, there's very little of it on the left, but you see much more purple on the right. Now it's hard to spot in the colorful plot, which is why we added the smooth signature exposures at the bottom, which is pretty much just looking at how much green do you see, and we just had a linear regression. And you see, indeed, there's lots of green on the left and it goes down, and purple and brown go up at the same time. There's also a plot in the middle, some unsupervised clustering, which I have completely forgotten why we added it. I think the reviewer asked for it and we just put it in. It's not showing very much. I think that might have been the point, because the problem with ovarian cancer is there are no distinct subgroups here. So this is a continuum of genomes, and that is what people, that's the kind of genomic chaos I described in the beginning. That's what people have been struggling with for a while. And the breakage fusion bridge part on the left and the HRD part on the right, that have been the only subtypes people had described so far. Whereas we now have a handle even on the 80% in the middle, and we can look at what particular mutational forces were active on these genomes. And that might be, and this is where my conflict of interest with a company comes in again, this might of course be interesting if you want to treat patients, because the biology of these different mutational signatures tells you something of what drugs you might want to give them. So for example, if you have a patient with a high HRD signature 3 or 7, you might want to give them a PARP inhibitor. Whereas if these signatures are not there, but the fold-back inversion is very high, you might want to give them a MEK inhibitor. So there is a direct, so instead of just seeing chaos, by looking at these signatures, we can dissect individual mutational processes which are active in a patient, and there is a potential therapy for many of them. Now, with that, let me find my water and take a little break while you guys digest that, and we get to the second part. Because everything I've told you so far was done in ovarian cancer. And it would be fair to say, well, I don't do ovarian cancer, what do I care? Which is why we did the obvious next step. So this is my student Ruben here. He's now Dr. Drews. He graduated almost a year ago, and he's still hanging out in the lab because we still haven't published this. I really hate reviewer 3 by now. What Ruben did in his PhD, he took the approach that we had pioneered in ovarian cancer and applied it to all of TCGA. And it's something that sounds very easy, and then you still spend 4 years on doing it. We have teamed up with Peter van Loo, who's a group leader at the CRUK and currently moving to MD Anderson, who's a worldwide expert in copy number analysis, hoping he would just solve all of this for us, and we still spend years on it. What I'm presenting you now, and that was actually in the title of the talk, his PhD now is a pancancer compendium of chromosomal instability. This is a systematic catalog of the extent, diversity, and origins of chromosomal instability. Will be a little bit of a cliffhanger because our patents haven't come through, so I can't tell you all the juicy stuff, but I will tell you as much as I can about this project. So the first plot, these are all the cancers in TCGA, around 33 or so. And the bars are the number of samples you find in TCGA. The whole number is bigger than 10,000. Every bar has a color, and for some bars you see a lot of gray on top of the color. We ordered them by the number of samples that have lots of copy number events where we think we can detect chromosomal instability in the genome. And the gray part of the bars, that's the number of samples where genomes were very flat and we didn't really see much chromosomal instability. And that is already interesting because in, for example, ovarian cancer, that's the cancer type on the far left, every sample has chromosomal instability. Whereas if you go to the thyroids on the far right, very large gray bar, very little purple, because these are very quiet genomes. So you see already there's particular cancer types which are very chromosomally unstable, and some are not so. But overall, it's about 80% of samples that show evidence of chromosomal instability. So it is a very widespread phenomenon in cancer. So what Ruben then did is the following. He took all the samples of TCGA, more than 10,000. We did some basic quality control to get it down to about 8,000 or 7,880. So I'm reading this now, the center from top to bottom here. And out of those, around 80% had detectable chromosomal instability. These were the colored bars on the last slide. For all of those, we derived absolute copy number. And then we used the type of approach I described, this kind of these global features and the NMF to derive signatures. And we did this in two ways. In the first approach, we just took all of the TCGA data together across all cancer types and derived signatures, and we got 10 of them. And then we thought, well, let's see what happens if we just take all the big cancer types with more than 100 samples and we derive signatures individually. And what we found is there were 7 other signatures which we had not uncovered in the pancancer analysis, but which happened repeatedly in the single cancer analysis. And we call them cancer type enriched copy number signatures. And if we add them up, the 10 pan-cancer and the 7 cancer type enriched, we now have 17 signatures of chromosomal instability. If you remember my cartoon from the beginning, that was the easy part, because this was the maths. You always get signatures, but do they mean anything? And this is where most of our work went in. So now we compared these 17 signatures with many TCGA covariates, from gene expression of DNA repair genes to appearance of extrachromosomal DNA, all the kind of weird genomic patterns people have described, contagious chromophripsis, all the signatures other people had done on the TCGA samples, and all kinds of germline and somatic mutations. That was not enough. So then we reached out to additional data sources, and luckily people had computed, for example, a method called HRDetect to detect HRD on many of these samples. We looked at the cell line projects from the Sanger and the Broad. We looked at Reactome, we looked at RNAi screens. So we looked at pretty much all the data we could put our hands on to understand what these 17 signatures mean. And I give you a snapshot of where we stand with this. The outcome of this is that we think we have now really taken down this umbrella term into its parts. We have defined the major drivers of sin. In particular, we have 3 signatures of homologous recombination, which is very interesting because usually people think of this as one unified concept, and we bring some more nuance to it. And we have survival predictors and new drug targets. And as I said, because our patents didn't come through, I can't tell you very much about the HR and drug targets thing, but I can tell you as much as I can about the other parts. Now, one of the reasons we integrated so much different data in here is because we tried to be as rigorous as we could be in understanding the biology of our mutational signatures. And our general strategy was like this. In my thinking, there's different types of chromosomal instability, different types of sin, and they are caused by different cellular processes. Now, a sin signature is a measurement or is a measure for one particular flavor, for one particular type of sin. In the same way that you often have particular marker genes for individual cellular processes. To give you a specific example, if we look at homologous recombination deficiency, that would be a type of sin. And the particular marker gene people look at would be BRCA, for example, BRCA1 or BRCA2. If you have a germline mutation in this, people think you're HRD, and that is a particular treatment decision. So the way we wanted to make sense of our 17 signatures is now in a 3-step process. First of all, we look at the signature patterns and we compare that to what other people have described how different types of sin look like. HRD cancers just look a particular way. That gives us an hypothesis for what particular cellular process is involved. And then we test this hypothesis by looking at the association between the activity of the SYN signature and marker genes of that process. So if we have a signature that's an HRD signature, we would expect that its activity is much higher in BRCA germline carriers than it is in people without BRCA mutations. And that's a test we can make. And then finally, if it passes that, we look at all the other data I showed you, at all the other individual additional evidence and independent data to strengthen that hypothesis if it's true. To give you an example, here is an overview of all the evidence we have for one of the signatures. It's called CX3. It's an HRD signature. What you see in the middle there, that was step one, the pattern of change. And the first thing we noted is the segment size is indicative of large-scale state transitions, which is one of the things that have been described for HRD cancers. And if we then look at the mutated genes, indeed, it's highly correlated with BRCA1 and BRCA2. And if we look at the orthogonal data, we see it's highly correlated with platinum sensitivity, single base pair substitution signature, which is for HRD, and actually 7 other HRD metrics which other people had published. So safe to say for us, this is an HR signature. I mean, we really tried everything there. And you can see on the left, there's some little yellow stars because we wanted to be very explicit and transparent in how much evidence we have for different signatures. Not all of them are as strong as this one. So you get a star, up to 2 stars if the pattern is correct, is interpretable, and it fits to the mutated genes that you see. And you can earn a 3rd star if you also got lots of orthogonal data. So with this, here's an overview of our 17 signatures. They are ordered from top to bottom by their pan-cancer prevalence. So signatures that appear in many cancers are at the top, and the ones that appear in few cancers are at the bottom. And you see many of them, so in the middle column, you see the stars. Many of them have 3 stars, so they look like the one I showed you on the last slide. Some of them we know nothing about. I mean, CX7, for example, appears in 50% of all cancers. It has a putative cause we don't know. There's a little bit we see in the pattern is segments with small change points, and that is it. So gives us very little to work on. Whereas others, for example, CX3 or CX5, the ones that's impaired HR. I mean, let me show you. There's some patterns in here. So the first group of signatures are mitotic signatures, and that's 1, 8, and 14, for example. The way you recognize them is because the pattern of change is very big events, capturing whole chromosome arms or whole chromosomes. And that's really something that happened during cell division when the pulling apart of the chromosomes went wrong. Another group of signatures is impaired HR, and we have 5 of them, 2, 3, and 5. 3 is the strongest one. That's why I showed it to you. It's the closest to previously described forms of HRD. The other ones are weaker. CX2, for example, we think is just some kind of background noise happening if you have any problem with HR, even if it is just very small. And there's other much more wider patterns. For example, replication stress is associated with lots of our signatures. It's kind of more a modulator that gets added onto other defects to be visible in the genome. Now with that, unfortunately, with this overview, I have to stop with this already, and I can only promise you there's more juicy bits. For example, the HRD signatures, it turns out, when you use them to predict platinum sensitivity, they're excellent for this and much better than other markers which have been previously described. And we also use these signatures in the cell line data where we had drug data available to see if there were biomarkers for particular drugs. And we have very promising results for that too. But I have to wait until the paper comes out to discuss that. Just to make sure you're not too disappointed, I'll end with some unpublished stuff, which we're currently working on. And I really wonder why my mouse is sometimes not working. Here we go. So what I'm currently working on and what I really try to get funded, so if you're one of the reviewers of my grants, please be nice. I want to better understand how to use these mutational signatures of SYN to guide treatment decisions. And I want to develop a pharmacogenomic test that tackles SYN at its roots at the mutational processes, and to bring some new biomarkers and drugs to cancers which have seen no progress for decades, like ovarian cancer. It's really terrible. So far, nobody's working at this. So if you want to do genomics, talk to me. So the key idea is this, and that's in a way a summary of the talk so far. If we observe a genome with high levels of chromosomal instability, my worldview is that underlying this are different mutational processes, the colored arrows, which are represented by different signatures, different fingerprints. So if I, from a patient's genome, figure out which mutational processes are active, then I have something to predict which drugs I could use for that patient. Now, in my future work, I will address the key limitation of everything I've told you so far. And I have not talked about it very much because it's such a fundamental limitation. Everything I talked to you about so far was based on bulk sequencing. And bulk sequencing tells you about the history of the tumors. In bulk sequencing, you see genomic events if they were part of clonal expansions and if they are shared by many cells. But it's a view of the history. I mean, it's when people do phylogenetics and cancer evolution, they want to do bulk, maybe, because they actually see the whole history. They do what people call archaeology, because they uncover the early events, but it's not very good at figuring out what is currently happening in a genome. So I try to indicate that in my colored arrows by saying the blue and the orange one, these are ongoing mutational processes. They happened in the past, but they're still happening now in the patient. So I would assume they're better to target than the green one, which happened in the past, left its marks in the genome, but now is extinct. So my future work will be on understanding the difference between ongoing mutational processes and those that happened in the past but have stopped. Now, the tool to do this is a single cell sequencing. And you see this in the right plot here, because the key idea is that ongoing SYN processes are visible in single cells because only the ongoing ones, but not the extinct ones, continue to add new mutations to the genome. So the ones which are extinct, they changed the genome already, and now these changes are just given from mother cell to daughter cell, but they're not changing anymore. Whereas if a mutational process is still ongoing, in every cell division, it has a chance to put additional mutations into the genome. So if I find a process that just keeps static, it's the same in all individual cells, this one is extinct. And if it is different across individual cells, that one is still ongoing. So the key methodological idea is, if we look at all the cells in the biopsy, then the shared events, that is the kind of traditional approach which I told you today. It's the same for bulk sequencing. Will tell you about certain signatures in the cells, but might miss some. For example, signature 4 is being missed here. But if we look at unique events, we see actually only the orange and the red one have unique events in single cells, which are indicative of ongoing mutational processes. So the idea is by going from bulk sequencing to single cell sequencing, and by counting unique events in single cells, we can actually distinguish extinct from ongoing mutational processes. Why is that useful? Well, if we now apply this test, and I plan to do this on cell lines, in organoids, and then in patients, and correlate this with drug treatment, we can, for example, figure out if ongoing and extinct signatures really have a different predictive power. Maybe they have not. It's an intuitive concept, but might be wrong. And we can also see how well they compare to many other drug predictors and biomarkers that other people have described for the same drugs. So I plan to work with the cell line compendium at the Sanger And there is large international consortia which try to predict sensitivity to drugs from these data. And I will just add another predictor and just see how well I do. We haven't tried it yet, so I'm still confident. So with this, my summary, please from now on, do not use SYN as an umbrella term. There are just too many flavors. Use one of the 17 flavors I've presented. What I've shown you was a systematic framework to measure these different types of SYN and to correlate them and to measure their impact on clinical phenotypes. What I could not show you, but what exists is that these SYN measures are very useful in practice. They can be used as biomarkers for new and also to identify new drug targets. And very specifically, the HRD signatures are excellent predictors of platinum sensitivity. There are lots of limitations to what I've presented. So here are the three main ones. Everything I've shown you so far was just single sample collections. So we cannot yet characterize changes in mutational processes over time during treatment or in relapse, because we would need multiple samples for that. Everything I showed you, except for the future work, which I haven't done yet, was done in bulk samples. And now that means it shows you the history of the tumor, but not necessarily what is happening right now. And most of the stuff I showed you, like all of the TCGA work, is based on SNP6 arrays. That's a microarray. And only the older people in the audience might still remember what a microarray is, because nobody's using this. So there is a question of translation because nobody's doing SNP6 in the clinic. So we have to come up with some sequencing approach. And we want to borrow from the ideas in ovarian cancer, where we use shallow whole genome sequencing, which is a very cheap way of doing whole genome sequencing, and to use these approaches. And that is exactly what this company is currently developing. So with all of this, thank you very much. This is a picture of my lab. So Ruben is I'm the guy who holds the biology of cancer on my lab, because as you can see, we read a lot. I mean, there's even one guy here who reads 2 books at the same time. And I'm having the biology of cancer in my hands because that's the one book I really should read for my job, but haven't. Ruben, whose work I described, is sitting on my left, and all the others are part of the different teams. When we submitted the paper, our figure 1 and proposed title was Pandemonium of Sin, but people didn't like, you see, pancancer, pandemonium, and then sin and sin, but the journal didn't like the dad jokes and the kind of stupid jokes, so they didn't go for it. So thank you very much for listening to me, and I'm taking questions.
Thank you so much for this exciting talk. So we have people in the in person in our Forest Hall, and we also have a number of people online. So for people who are online, you can unmute yourself and ask the question yourself. And for the people in person in our forum hall, I think it's really difficult for me to see you. So probably you have to talk with Aaron, maybe come to the podium and ask a question somehow. Okay.
That is not super crowded, huh?
No.
Well, we're still.
I have a question. Gil Oman here.
Yes, please.
Okay. First of all, thanks for a very substantial and lovely presentation. It's tremendous work. I'm part of the CPTAC consortium, the Clinical Proteomics Tumor Analysis Consortium in the US and globally. And I'm quite keen. I see you made very good use of TCGA data. I'm not sure I saw all the slides. I had an interruption. So I want to ask you if you use the proteogenomics data and now published in a stream of papers in Cell, including work on ovarian and many other cancer types.
Oh, so you caught me out there, right? When I said we used all the data was available, and now the project, I don't think we did actually, and we should, because I think what it could tell us is the activity of DNA repair pathways. And that's, we currently get this out of gene expression, but you're completely right. We should just look at the proteomics data.
Right, and it would be a nice parallel to your work, because we do try to perform pathway analyses, and of course PTMs of proteins you cannot predict from the
That's right.
DNA and RNA. And we also recommend targeted therapies as appropriate.
Yep. That makes a lot of sense. Yep. Yeah, we should definitely look into that.
Great, thanks.
Hello. Can you hear
Hello.
me? Hi. Thank you very much for your presentation. I wanted to ask about the first part of the presentation about the signature study that you had on ovarian cancer. I'm wondering if you have tested any of those signature on whether they have a power to detect the platinum sensitive versus refractory in ovarian cancer with the already published work. And I'm wondering whether that's because it's something that you have already published, whether we can use that for other ovarian cancers to detect the sensitivity versus refractory.
That would be signatures 3 and 7 in the original publication. So that is part of the, I think it's 2018 Nature Genetics. That should be part of this. The other one is even better. And I hope you don't have to wait for too long before we just have to fight down this one reviewer, and the other paper will be out too. So we really did the pancancer work, really improved the signature definitions and the resolution we could get. I mean, you can definitely right now try out the original signatures, but just wait a couple of weeks, hopefully, and you get the better set.
Thank you.
Florian, thanks. I thought that, you know, at that slide, you would be the guy writing the book instead of reading it. Maybe that's what you got to do the next time. So thanks for a lovely talk. Like Gil, I really enjoyed it a lot. And I saw a few slides where you had the telomeric length as a covariant. And I wonder if you could expand on that a little bit. Have you found any correlations or causalities or anything there that we should know about?
So the telomere length is a key indicator of the or key piece of evidence for the breakage fusion bridge signatures, because so the mechanism really is so the telomeres get very short, so the ends of the chromosomes stick together, and when they get pulled apart, it doesn't really break at the same point again, and you get these overlays at the end. And this is where they really come in. So in the ovarian cancer, this is why they were so prominent for signature 1. And we found them again in pancancer as one of our, it's actually, again, I think signature 1. It's a very, very widely visible pattern in there. But it's also very specific in a way, which is why it's very useful for us.
Yeah, thanks a lot. It was kind of my impression. I appreciate you expanding on that a little bit.
Thank you.
May I also ask a question? Since you read a lot, I'd like to ask a question related to literature. So when I was in graduate school, I was really fascinated by the paper called Hallmarks of Cancer.
Yep.
I believe there's one, the first one in 2000, and there's another one 10 years after. And they listed all the hallmarks in different categories. So since this almost 10 years, after 10 years of their second paper, I wonder, from your discoveries, do you feel like what do you find agree with what they suspect 10 years ago, like all these different hallmarks?
Oh, that's a big question. It's almost a philosophical question now. I mean, there's only more and more hallmarks, right? I mean, I don't think they lost any on the way. I mean, in the first paper, they had 6 or so, and now we're at 10 maybe.
Yes.
It's more and more of them. I think one of the main changes over time was people realized it's not just cancer cell intrinsic effects, but it's also the tissue microenvironment, the immune compartment. There's lots of other things surrounding the cancer cells. In a way, my own research is very limited in that respect. I mean, we are very much team DNA here, and we look at cell intrinsic drivers of cancer. So I'm really interested in the cancer cell, what is going wrong there? And this will have an impact or some kind of connection to what's happening outside the cancer cells, but we have not really started exploring that yet. So there is big chunks of the hallmarks that I'm a bit unconnected from still, because currently our research was just trying to measure some of these processes in the cells much better. So chromosomal instability, I'm pretty sure, is one of the initial hallmarks, or at least something very close to that. So in a way, then our own work is only taking this hallmark and bringing more nuance to the discussion and having more quantitative measurements of a term which has already been described in this field for a very, very long time.
Okay, thank you so much. It seems right now your research is more on the fundamental cellular processes, and their hallmarks is more like global view of all the.
Could be, yeah.
Great. Do we have more questions from the audience? Oh, I think there's one.
Something in the chat, huh?
Yes, it's from our director of our genomics core, Professor Wilson, Tom Wilson. Are structural variants with endpoints in fragile site genes, very large genes, a pervasive feature of all replication stress signatures, or restricted to a specific subset of those signatures?
Yeah.
Didn't have a mic at present, so that's a great question, and thanks, Tom, for asking. You're on here. You know, he's on the Zoom too.
That is a great question, and I don't know the answer to it. Because the replication stress signatures, there's not a specific one for replication stress. It's somehow, it's usually, we see a certain, it contributes to many, but we usually see there's many other reasons why we have chromosomal instability, which are being associated with the signature, and then on top, replication stress. So this is, for example, for the HRD signatures, I told you we have 3. The first 2 don't have, the first one doesn't have any replication stress, and then only the second one, which is stronger, has the first kind of pattern plus the replication stress associations on top. Now, I have not looked at it from this perspective of the fragile site genes, the large genes, so I cannot really answer that. Would have to look into the data again.
Thank you.
Okay, I don't see more questions. Thank you again, Florian, for this wonderful talk. Thank you so much for spending this 1 hour with us. We learned so much from you, and we look forward to meeting you again sometime. Thank you.
In person,
Thank
too.
you very much. Thank you. Yeah, hopefully next time. Good. Thank you.
Well, we hope there is a next time. Thanks so much. Delighted to meet you.
Thank you. Bye-bye.
Bye.
Bye, everyone. Bye. Thank you.